This in vitro disease model can be used to study the interaction of whole blood and patient-derived cells, specifically the endothelium, to evaluate thrombogenicity and the effectiveness of anticoagulative agents.
This system with primary endothelial cells can be used under various circumstances and can be modified by introducing custom-made microfluidics or by adjusting blood flow parameters.
In addition, this model can be used to study thrombus dynamics in inflammatory conditions or to assess the effects of anticoagulant and antiplatelet therapy. Ultimately, these approaches can be used in a method of personalized medicine.
After obtaining a surgical human pulmonary artery tissue sample, coat a high-affinity cell-binding 60 ml cell culture dish with 2 ml of 5 micrograms per milliliter fibronectin and incubate the dish at 37 degrees Celsius for at least 15 minutes. At the end of the incubation, use sterile forceps and a laminar flow cabinet under sterile conditions to transfer the tissue into a petri dish of PBS. If the tissue is still in a ring, cut the artery open with scissors, taking care not to touch the innermost layer of the vessel, as the endothelium is easily damaged and removed. After removing the fibronectin, add 4 ml of complete endothelial cell medium to the dish, and use forceps to transfer the tissue sample into the medium. Use a scalpel to carefully scrape the inner layer of the vessel into the medium, taking care to avoid any lipid accumulations that are observed in the vessel wall. After the cell